So it is my pleasure today to introduce Isha Jain. She's an assistant professor at the Gladstone Institute in UCSF. So her work touches on something that is of interest to not just people in the department like Alessandro, but also myself, which is very interested in understanding the role of oxygen, hypoxia, and normoxia in mitochondrial function. She did actually some really cool work. And I remember when these papers came out, like looking at hypoxia and how they're protecting against mitochondrial diseases and stuff like that. And she has some really nice papers that kind of go into the molecular mechanisms and how that has been or how that's worked out. And she did all that. You were in graduate school at the time,
Mostly, yeah.
In Vanti Mutha's lab, and then after that, she moved to UCSF as a faculty fellow for a couple of years, and then just recently, in the last year, started her own lab. So, where I am assuming you are going to continue your work on this really cool hypoxia story in mitochondrial diseases. So, please take it away.
So thank you so much for having me. I'm excited to tell you guys a bit more. I think this was rescheduled once or twice, so thanks for being flexible. Yes, it's my pleasure to virtually be there. I'll tell you today about my lab's research program focusing on what we call turning the oxygen and the vitamin dials. So broadly speaking, my lab is really fascinated by the idea that we can change things about the way we live, so what we breathe and what we eat, as therapeutic strategies. So rather than thinking only about small molecules or drugs, can we change things about our physiology as potential therapies? So I always start with a little bit of a nod to the past. I'm fascinated by the history of science. And so since my lab studies oxygen, we've been reading a lot about the first oxygen scientists. And so it was actually pretty difficult to study the effects of hypoxia until about 150 years ago. And what happened was at that time, hot air balloons were invented, enabling researchers to ascend very rapidly to high altitudes. And of course, at high altitude, there's less oxygen, and so they could study the effects of hypoxia. And so these were the first aeronauts, as they were called. And they would ascend in these hot air balloons, and they'd have a cage of pigeons, and they would release pigeons at different heights and observe their physiology, see what happens to their flight, their behavior, etc. And of course, at a certain altitude, the pigeons would not do so well. And if the pigeons didn't do well, by similarity, of course, the investigators also did not do well. And so this photograph is actually of a pretty horrible accident that happened where this hot air balloon ride called the Zenith, these three aeronauts ascended to 20,000 feet over the course of about 30 minutes to an hour. And that's very fast, and humans cannot adapt that fast to hypoxia. And so actually two of these three scientists actually passed away in this flight. So, people have been passionate about studying oxygen for quite some time, but luckily now we have much safer ways to study hypoxia physiology and metabolism in the lab. And so we have hypoxia chambers that don't require hot air balloon rides. So today I'll tell you a lot about the story that Scott mentioned, thinking about how we can turn the oxygen dial to hypoxia or hyperoxia as a therapeutic. And then I'll touch about some of our more recent work and future plans, thinking about vitamin deficiencies and vitamin therapies. And so those are separate arms of the lab, but with some overlapping questions in the middle. Okay, so the big picture questions that we're asking is, number one, why is too little oxygen toxic? And of course, this has been studied for quite some time now because low oxygen is involved in 3 of the 5 leading causes of death in developed nations. So heart attacks, strokes, respiratory failure. And so in all of these conditions, low oxygen and low nutrients are implicated in the pathology. But it's still unclear exactly which oxygen-dependent reactions fail as a function of oxygen. And so we're doing a lot of systems biology approaches to understand that. What we're equally, if not more interested right now is understanding why is too much oxygen toxic. So it turns out that if you take any organism, more or less, and you put them in 100% oxygen, they die within days. And this is actually much less studied. What happens when there's too much oxygen? Typically we get the reactive oxygen species type of answer, but we're more interested in figuring out exactly which molecular pathways are perturbed in high oxygen. How does the body sense variations in oxygen levels? And of course, that was the topic of the recent Nobel Prize about HIF biology. But HIF has a negative feedback loop. And so are there chronic hypoxia sensors in the body? And what are the mechanisms at play? How does the body adapt to variations in oxygen levels? What are the diseases that are characterized by too little or too much oxygen? And so beyond the canonical ischemic disorders, what other conditions are characterized by this imbalance in oxygen supply and demand? And then what we've really been pushing is how can we turn the oxygen dial to low or high as a therapeutic strategy? So simple intervention that could potentially affect disease progression. And then we're applying this exact same framework to vitamin and cofactor biology. So very similar principles, but thinking about oxygen as a metabolite or vitamin cofactors as metabolites. So today I'll tell you about sort of recent past work about how we came to develop hypoxia paradoxically as a treatment for mitochondrial disorders. And then I'll tell you about how we're thinking about these questions of matching metabolic supply and demand for vitamins. So before I get into that, evolution has really come up with beautiful ways to match oxygen supply and demand. And so there's elaborate mechanisms in place to ensure that tissues get enough oxygen. So things like red blood cells and hemoglobin binding oxygen, our entire cardiorespiratory system. And here on the left is a picture of E. coli performing aerotaxis. So this is a bubble of oxygen. And so E. coli can actually sense the amount of oxygen in their environment and swim away or towards it. And turns out that's a principle that's common to multiple organisms. But what we would argue is equally important is the ability to avoid oxygen when it is in excess. And we like to claim that the mitochondria are the primary oxygen detoxifiers in the cell. So the mitochondria consume 90% of oxygen in the body. And so it's a way to maintain low oxygen tensions within our cells and our tissues. And we'll talk about why we think that's important. But also, a lot of our gut microbiome exists in extremely hypoxic niches. Stem cells tend to be in hypoxic areas of the body. So it's clear that the body has evolved to maintain low partial pressure of oxygen in certain situations. So how did this story start? I wasn't really thinking about oxygen at the time. Instead, I was focused on mitochondrial disorders. So mitochondrial disease, as you may know, is a devastating class of diseases. It affects about 1 in 5,000 live births. It's the most common inborn error of metabolism. And nearly every organ system is affected, resulting in blindness, deafness, muscle myopathy, neurodegeneration, etc. And it's a really challenging class of disorders to target because of this multisystemic effects and the underlying variability in the different mutations that cause mitochondrial disease. And so currently as it stands, there isn't a uniformly applied therapeutic strategy beyond sort of vitamin cocktail mixtures. And so I wanted to tackle this problem more systematically and ask genome-wide, what is the ideal therapeutic target for mitochondrial disease? So if we had to perturb one gene or gene product that would alleviate mitochondrial dysfunction, what would that be? And so this is early days of genome-wide CRISPR screens, which is one of our bread and butter methodologies. And to make a long story short, we exposed cells to a mitochondrial toxin or electron transport chain toxin, antimycin, and asked out of all of the genes in the genome, if you knock out every single gene, which knockout enables cells to survive in the face of mitochondrial dysfunction. And so here are the results of that genome-wide screen. Every dot is a different guide RNA knocking out a given gene. So 20,000 genes, 5 guide RNAs per gene. On the x-axis is the rank of the guide RNA and its ability to rescue disease. And on the y-axis is the effect size, essentially. So how well could that knockout rescue mitochondrial dysfunction phenotypes? And it turns out that we had only one very strong hit. And so all 5 red dots here correspond to the exact same gene, which is VHL, von Hippel-Lindau factor. And so that's a pretty strong hit for a genome-wide CRISPR screen. So out of 80,000 guide RNAs, knocking out this VHL gene resulted in some of the highest ranks of the ability to rescue this mitochondrial dysfunction phenotype. And in this setting, we're using cell survival or cell growth as our readout. So what is VHL? We first looked this up and it said von Hippel-Lindau tumor suppressor. And I panicked because I said, oh no, knocking out a tumor suppressor does not seem like the best therapeutic strategy. But we pushed forward, and I'm glad we did, because it led us to the world of oxygen biology. Okay, so I'm telling you that knocking out VHL seems to be protective in cell culture when cells are exposed to this mitochondrial electron transport chain toxin. So let's quickly review where VHL is in the world of hypoxia biology. So the hypoxia-inducible transcription factors, or HIF proteins, are constitutively made in the cell. And then under high ambient oxygen tensions, or normoxia, the perolyl hydroxylase enzymes take this oxygen, hydroxylate these transcription factors, and then it's specifically this hydroxylated form that is recognized by VHL, which is an E3 ligase, which ubiquitinates the HIF transcription factors and results in their degradation. So under normoxia or high oxygen, this protein is made, degraded, made, degraded. So in hypoxia, on the other hand, you don't have oxygen, atmospheric oxygen levels. And so this cascade does not take place. Instead, HIF is stabilized, it enters the nucleus, and it turns on the transcription of several hundred genes that enable survival in a hypoxic environment. So things like rewiring the TCA cycle, increased glycolytic flux, increased hematocrit, erythropoietin, et cetera. And so what happens if you knock out VHL in our screen? So it turns out if you knock out VHL, VHL is the negative regulator of HIF. And so you're artificially activating the HIF response even under normoxia, right? So you're triggering this adaptive pathway even when there is no signal for low oxygen in this setting. And so we saw this and we thought, okay, that sort of makes sense, right? Mitochondrial dysfunction is a form of impaired oxidative metabolism, right? But it's a type of impaired oxidative metabolism that's not evolutionarily been selected for. And instead, hypoxia is also a form of impaired oxidative metabolism, but evolution has solved the problem by creating this adaptive pathway. So maybe we're borrowing this solution from hypoxia and repurposing it for mitochondrial dysfunction. So we kept going and thinking, okay, that might make sense. But we wanted to turn this into a therapeutic strategy. And so we asked, are there any small molecules that have the similar effect of activating the HIF response in normoxia? And it turns out there's an entire class of drugs that are being developed to inhibit the PhD enzymes, which are also negative regulators of HIF. And so these drugs have the same effect of activating the hypoxia response, even when the signal is not present. And so we thought, okay, let's start working with this compound to see if we can have the similar rescue of mitochondrial dysfunction. And so we began using this drug that activates HIF in normoxia, and we took K562 cells or 293 T cells. We gave them mitochondrial toxins, so complex 1 inhibitors, complex 3 inhibitors, or complex 5 inhibitors. They all impair growth in red. Now, if we just co-treat with this HIF activating drug, you see this pretty remarkable rescue in blue back to almost normal levels in black. And so what we showed here was that just regardless of how you disrupt the electron transport chain, complexes 1, 3, or 5, activating the HIF response is protective in cell culture. This was exciting, multiple types of mitochondrial disease were being rescued by our therapeutic approach. But, again, we really wanted to take this all the way in vivo. And so we spent quite some time trying to get this drug to work in the premier mouse model of mitochondrial disease. And so this is a mouse model that I think many here have worked with. It's a knockout of a complex 1 subunit, NFS4, and it mimics the most common pediatric mitochondrial disease known as Lee syndrome. And so these mice develop these T2-weighted symmetric lesions that are detectable by MRI in the vestibular nuclei, very similar to the human Lee syndrome patients. And these mice have a very similar presentation to the human patients as well. They have a body weight loss, they're blind, they're deaf, they have locomotor defects, and ultimately they succumb to respiratory failure. And so we thought we would start by testing this drug in these mice. And I actually lost probably almost a year of my life trying to get this drug to cross the blood-brain barrier. Turns out this drug was actually being developed for a completely different indication, renal anemia, and so it was optimized for the kidney. And so I was trying to get this drug to somehow reach the lesions, and it wasn't working. And then finally we had this moment. It was sort of a last minute experiment. I had a friend across the street who was working on hypoxia for another purpose. And I thought, okay, I'm struggling to activate the HIF response. What is the best way to activate the HIF response? Well, it turns out it's probably just putting the animals in hypoxia. And so this is sort of a late night experiment where we snuck a cage of these NFS4 knockout mice into these hypoxia chambers and just left them there thinking, let's see what happens. And that was really the eureka moment for this project. And so I think the best way to show this is to just show you what happens to these animals in hypoxia. So these are 5 knockout mice. They're all knockout for NFS4. They're all siblings. The only difference is that the 3 in the middle have lived in normoxia or room air, 21% oxygen. And the other 2 in the corners have been living at high altitude equivalent. It's about 5,000 meters equivalent, which is about 10% oxygen. So sea level is 21%. The 2 in the corner have been at 10%. And so this is a really black and white result. So the animals in the middle who've spent their life at sea level are blind, they're deaf, they're probably hours away from death, whereas the other 2 who are genetically identical but have been living at high altitude seem indistinguishable almost from wild type animals. And when we initially published the story, all of the sea level treated knockout mice had died by about 2 to 3 months of age. And at this time, all of the hypoxia treated animals were still alive. So it turns out they lived to about 5-fold longer, about a year of age, but this really remarkable therapeutic effect or extension of lifespan. And we could rescue nearly every aspect of disease. And so here is body weight of these animals. So in red here are the knockouts at room air who lose weight and die at about 2 months of age. And hypoxia rescues us at least partially in blue. We can measure body temperature in these NFS4 knockout mice. It's decreased in the knockouts in red at sea level and rescued by hypoxia in blue. We can also perform various behavior tests like their ability to stay on a rotating rod. Again, this is impaired in the knockouts at sea level in red and rescued by hypoxia in blue. And so I'm not showing the other examples, but nearly every aspect of disease was rescued from biomarkers to behavior to neuropathology. But this initial finding led to a whole range of additional questions. And so after that, we spent the last few years chipping away at trying to understand what's going on in more detail. So something that we're still sort of piecing away at is what is the mechanism of hypoxic rescue? Why is low oxygen having this really dramatic therapeutic effect? What is the underlying pathophysiology of disease at different oxygen tensions? So at room air, they die of this neurodegenerative disease, and is it the same cause of death in different oxygen tensions? Can we reverse disease in addition to preventing it? And so patients typically present after they've already had their first metabolic crisis. And so can we reverse the lesions in addition to preventing disease? How do we actually get this to reach patients? Can we develop a gas-free therapy that's more practical than living in the mountains? And then importantly, which additional disorders may benefit from hypoxia therapy? And so are there common conditions or other monogenic diseases that might benefit from low oxygen? So first, we began asking what is the mechanism of rescue and the underlying cause of pathogenesis. And our first clue came from a very simple experiment. So I told you that turning the oxygen dial to low has this remarkable therapeutic effect and extends lifespan by nearly 5-fold. So now what if we do the opposite experiment, which is very mild hyperoxia? So 50% inhaled oxygen, that's benign in wild type animals. So wild type animals can live in this oxygen tension for up to 2-3 years. But what happens if we take these NFS4 knockout mice and put them in moderate hyperoxia before disease presentation? So it turns out they die within a matter of a few days. So this extreme sensitivity to oxygen, where very moderate hyperoxia is fatal, normoxia results in 2-month lifespan, and hypoxia, they can live for about a year. And so this really pointed us to think more and more about oxygen toxicity and oxygen itself. And one very important takeaway from this, from a clinical perspective, was that we should perhaps be a bit careful about using supplemental oxygen in mitochondrial disease patients. So when it's clinically indicated and there's respiratory issues, maybe it makes sense, but perhaps otherwise we should at least titrate the amount of oxygen that we give mitochondrial disease patients. And actually, more recently, there's been a fair number of clinical studies showing that hyperoxia or moderate hyperoxia can even be pathological for non-mitochondrial disease patients following a heart attack or a stroke, for example. So oxygen is not always good, is the takeaway. So we're thinking about this in 2 different buckets. So either low oxygen is a signal that triggers adaptive mechanisms, enabling mitochondrial disease organisms or cells to survive. So things like the HIF pathway, or alternatively, it's more about molecular oxygen itself damaging certain biomolecules. So we began by testing the concept that is the HIF pathway that I talked about earlier sufficient to rescue disease in vivo, right? So we got to this whole concept by studying a VHL knockout cells. And so we asked, is HIF really the story in vivo as well? And actually, surprisingly, it turns out it's not the case, somewhat terrifyingly. And so here's a reminder of the HIF pathway that I had described earlier. And so what we did was we crossed the NFS4 knockout mice with knockout mice that lacked the PhD enzymes. So there's 3 of them, PhD 1, 2, or 3, or VHL, which is analogous to our genome-wide CRISPR screen. And so in all of these mice, we're supposed to be activating HIF in normoxia in the NFS4 knockout mice. And so the double knockouts are shown here in red, and the single knockouts, the NFS4 knockouts, single knockouts are in black. So not only do we not rescue disease, we actually make things even worse. And when we did MRIs of these mice, lesions appeared in regions that they didn't even exist in the single knockouts. So basically, HIF is not sufficient to rescue disease in vivo. It might even make things worse. And what we think is happening here is that part of the HIF response is to increase oxygen delivery, right? So increased vascular density, increased hematocrit, increased erythropoietin. And so you might actually be increasing the local partial pressure of oxygen in these mice by activating this HIF pathway. And so if HIF is not the main player here, then it might actually make things worse. Okay, so if it's not about HIF, then what is it about, and what's happening to oxygen itself in these animals? So we took the knockout mice and put them in these clams chambers, measuring whole body oxygen consumption. And the knockouts are in red, wild types are in black. And on the y-axis is whole body oxygen consumption, on the x-axis is age. And so as you can see, as the animals age, they have a decreased whole body oxygen consumption, which makes sense because they have a mutation in the electron transport chain, right? And as I told you earlier, the mitochondria consumes almost 90% of oxygen in the body. And so if you have a mutation in the electron transport chain at the whole body level as well, you're not consuming as much oxygen. Okay, so that makes sense. But what does that mean about the partial pressure of oxygen in the tissues of these mice? So they're breathing the same amount of oxygen, right? So the supply is the same, but the demand has now gone down because of a broken electron transport chain. And so then we can measure the amount of oxygen in the brains of these mice. On the x-axis here is the inhaled oxygen tension. On the y-axis is the partial pressure of oxygen in the brains of these mice in the vestibular nuclei. And so we can take a Clark electrode and measure oxygen directly in the tissues. And let's start here in 21% oxygen or sea level. What we can see here is that knockout mice have double the amount of oxygen in the brain, right? So at the same inhaled oxygen tension, there's double amount of oxygen. And by breathing hypoxic air, they're essentially normalizing this excess oxygen and bringing it back down to wild type levels. And so this is what has led us to think that perhaps this is one of the first diseases of hyperoxia, where tissue hyperoxia is a primary contributor to disease. And so a lot of what my lab is doing now is trying to understand what happens when you have excess oxygen in the brains or of any tissue of mice or other organisms. And so as an aside, people always ask what's happening. Is this about superoxide and reactive oxygen species? So one thing is that we can isolate mitochondria from the knockout and wild type mice. And this was done with Alexander Galkin. And we can measure the hydrogen peroxide produced by the mitochondria as a function of local oxygen tensions. And you can see here that, at least in this experiment, there's really not that much of a difference between knockout and wild type animals as a function of dissolved oxygen. However, as I did mention, the knockout mice do have more oxygen in their tissues, so that might be sufficient to increase the local amounts of hydrogen peroxide or superoxide. So we think that that might be part of the story, but that's probably not sufficient to explain our phenotypes. Okay, so what is the underlying pathophysiology of disease at different oxygen tensions? So I told you that in sea level or normoxia, the animals are dying of these lesions in the vestibular nuclei. And so that's in red here in normoxia. And so this is a full survival curve. But what's happening in hypoxia or in hyperoxia to these animals? And so we can perform MRI scans right before the animals die at these 3 different oxygen tensions. And so in normoxia here, you can see that there's lesions in the olfactory bulbs, these white dots here, or the vestibular nuclei as expected. But interestingly, in hypoxia and hyperoxia, there are no such lesions, suggesting that perhaps it is not a neurodegenerative disease in hypoxia and hyperoxia that is the main cause of death. So what we noticed in hypoxia, so the animals eventually did die at about a year of age, and they seemed to almost drop dead. So it wasn't this progressive weight loss that we saw in normoxia, but they abruptly seemed to drop dead. And so that made us think of more of a cardiac phenotype. And so we performed echoes of wild type and knockout mice that had been treated with hypoxia chronically. And we found that the knockouts did have impaired cardiac function, and this could be measured through various different parameters. And we even observed slight arrhythmias in the knockouts exposed to hypoxia. And so we think that in hypoxia, more of a cardiac phenotype might be relevant as a cause of death, though we haven't gone a lot deeper into that phenotype. Now, in hyperoxia, what was known is that if you take a wild type animal and put it in 100% oxygen for a few days, it develops really dramatic pulmonary edema and dies of respiratory failure. And so we thought, what's happening to these knockout mice in moderate hyperoxia? Is it the same thing? And so this is HND for these knockout mice and wild type mice in moderate hyperoxia. And you can see this dramatic pulmonary edema in the knockout mice. So in summary, what this tells us is that in 3 different oxygen tensions, these mitochondrial disease mice are dying of 3 different causes: neurodegenerative in normoxia, perhaps cardiac in hypoxia, and definitely pulmonary edema in hyperoxia. And so this is what got me really excited about trying to understand tissue-specific pathologies and thresholds for oxygen toxicity, which is a lot of what our lab does right now. Okay, but this whole time I've been telling you about how we can prevent disease by placing the animals in hypoxia before they develop disease symptoms. So then we wanted to ask, can we reverse disease in addition to preventing it, right? That would be a much more viable therapeutic option. So here is body weight of these knockout mice. So if we leave them in normoxia in yellow, they lose weight and die. We can place them in hypoxia before disease starts in gray, and they gain weight and do well. Now we can start a different cohort where we leave the animals in normoxia, wait until they're at the brink of death, and then turn the oxygen dial to low. And there's this remarkable reversal of disease phenotype. So within even 2 or 3 days, the body weight starts to become rescued in these animals. And perhaps most excitingly is that we can wait until these animals are extremely sick, have these 2 lesions in the vestibular nuclei, then we can turn the hypoxia dial and the lesions disappear. So we've now done this with even more extreme hypoxia, and they disappear in about 1 week of hypoxia exposure. So simply living in hypoxia can reverse the neurological lesions in these animals. So that's exciting. We don't know exactly what that means. What aspect of the lesions are we rescuing? Presumably we're not, bringing cells back from the dead, but perhaps edema or some sort of immune response is being rescued. Okay, but how do we turn this into a viable therapeutic strategy, right? So we can make our houses hypoxic. So it turns out that there's equipment available to make living spaces hypoxic that you can buy for $1,000. And this is primarily used by athletes who are trying to mimic altitude and train sort of in the mountains, essentially a form of doping. And so that's actually the equipment that we use in the lab to place the mice in hypoxia. So we can do that, but can we think of other creative ways to cause tissue hypoxia? So we've had fun coming up with sort of crazy ideas here. So as I mentioned, living in the mountains is one option. We could, decrease oxygen delivery to the brain through surgical methods, not advisable. You could just decrease the amount of red blood cells through essentially bloodletting. And that sounds crazy, but people with polycythemia actually do get that as a treatment regularly. It turns out low-dose carbon monoxide competes with oxygen for binding hemoglobin. So theoretically, that would cause tissue hypoxia, and that's actually been used for other indications in clinical trials, early-stage clinical trials. But all of these are sort of crazy ideas that probably no parent would agree to. The most viable option that we've been thinking about is using drugs that affect the hemoglobin binding affinity for oxygen. So left shifting drugs that basically increase how tightly hemoglobin binds oxygen, preventing it from being released in the tissues. And so we're working on trying to optimize that as a potential therapy. But in the meantime, as a proof of concept, on the left here is the oxygen delivery equation. And so you can affect the amount of oxygen in the tissues of organisms by changing any of these variables, whether it's anemia, carbon monoxide, beta blockers, left shifters, etc. And so just for fun, as a proof of concept, we treated the knockout mice with low-dose carbon monoxide or anemia. And you can see that all of these interventions extended lifespan pretty dramatically in red compared to untreated mice in black. So just the proof of concept that it's not just about inhaled oxygen tension, but anything that perturbs oxygen delivery has this potential therapeutic effect. And then the next question that we've been tackling is which additional disorders may benefit from hypoxia therapy. So this whole time I've told you about a very specific mutation, NFS4. Is this relevant for other types of mitochondrial disease and perhaps even more generally? So to answer this, we performed a genome-wide CRISPR screen in normoxia and hypoxia. And this was not just about hypoxia therapy, but more broadly, what are the mechanisms of hypoxia adaptations? And of course, this is relevant to a whole range of additional diseases. So we took cells, placed them in 3 different oxygen tensions, and we created a genome-wide pool of knockout cells and asked which knockouts survive better in hypoxia than normoxia. And we did this genome-wide, hoping to uncover comprehensively which diseases can benefit from low oxygen. And so we can do pathway enrichment analysis. And as expected here in red, the category of genes that are rescued by hypoxia include oxidative phosphorylation or electron transport chain, and specifically complex 1. So at least in this data set, complex 1 seemed to be the primary disease category that was rescued by hypoxia. Interestingly, on the other side of the coin was a completely different organelle. So peroxisomes seem to be selectively more essential in hypoxia and less so in normoxia. And I won't go into the details here, but that brings up the idea that perhaps we can turn the oxygen dial in the opposite direction for peroxisomal disorders. But now zooming into the knockouts that were rescued by hypoxia, we identified 75 disease genes that were rescued by hypoxia in cell culture. And so they're listed here. So in dark red here are diseases that are directly known to impair electron transport chain function. And you can see a lot of NDUFs, so a lot of complex 1 disease genes. In light red here are indirect causes of electron transport chain deficiency. And so these are monogenic disorders, but they're not mutations in the electron transport chain or assembly factors. And then black here are non-mitochondrial disease genes. And so this leads to the exciting possibility that perhaps many additional disorders can be rescued by hypoxia. And so we've been chipping away at this list and seem to be validating at least several of these disease genes with hypoxia therapy in vivo. I'm going to skip the peroxisomes for a bit in the interest of time. So since then, we've started phase 1 clinical trials over at MGH. This was just done with a hypoxia tent, as I mentioned, using the equipment that's used by athletes. And of course, people do fine in the phase 1 trial because people have lived at altitudes for quite some time. And hopefully moving forward, we can move this towards a small molecule approach. So in summary for this portion, I've told you that hypoxia extends life of mitochondrial disease models by about 5-fold. We can reverse disease, not just prevent it. Turns out that activating the HIF response is actually not sufficient to rescue disease. So even though we started this project thinking about HIF in cell culture, it turns out in vivo, that's not the primary mechanism. Instead, we think it's a disease of hyperoxia, so elevated partial pressure of oxygen in the brains of these mice, we think is a primary cause of disease. We can come up with creative ways to decrease tissue oxygen levels. And now we've nominated 75 or more monogenic disorders, and a lot of my lab is working on some common conditions as well that can be rescued by low or high oxygen. Okay, so I've told you about how we're turning the oxygen dial as a therapy. Low oxygen is protective for mitochondrial diseases. Perhaps high oxygen is protective for peroxisomal disorders, though we're still thinking about that. But this led us to think more generally about how can we change simple variables in our environment as therapeutic strategies. And so now we've been thinking a lot about applying the same framework to vitamin and cofactor biology. So how can we change the amount of vitamins or cofactors in our diet as therapeutic strategies? So again, starting with a bit of history of science, from the late 1800s to the 1950s was this really beautiful era of rich biochemistry when the 13 vitamins were discovered. And this led to over a dozen Nobel laureates who discovered each of these vitamins and then discovered their specific metabolic role in biology. And then something happened around the 1950s, maybe molecular biology happened. And I'm saying this kind of tongue in cheek, but vitamin biology entered this sort of pseudoscience realm where people started taking sort of random mixtures of vitamins and megavitamin therapy without sort of a scientific rationale necessarily. But it turns out that specific vitamins do have really dramatic disease ameliorating effects for very specific diseases. And so there's countless clinical reports, for example, showing that vitamin B3 or niacin has lipid lowering properties. Now there's a lot of work being done on lifespan extension, et cetera. And to date, there are over 5,000 clinical trials ongoing using different vitamin cofactors as therapies. But in most of these cases, it's not based on a specific scientific framework, but rather on sort of anecdotal stories of individual patients being helped by a given vitamin. And so we wanted to tackle this using our sort of more systematic approaches and think about exactly which diseases can benefit from each of the different vitamins and cofactors. And of course, vitamin biology is relevant more broadly. So for example, 250 million preschool children have vitamin A deficiency resulting in blindness. Much of the elderly population, even in developed nations, has subclinical vitamin deficiencies. And despite adequate dietary intake, there just isn't enough gut absorption of a lot of the different vitamin and cofactors, resulting in sort of subtle phenotypes. And this is actually relatively understudied, but we think that subclinical vitamin deficiencies, meaning not enough that you present acutely in the ER, but sort of just below sort of required amounts of vitamins, are linked to a lot of common conditions like cardiovascular disease, diabetes, et cetera. And so there's a lot of work to be done to systematically understand how variations in vitamin levels can affect these different types of conditions. So the questions we're asking: which diseases can be treated by megavitamin therapies? So which monogenic disorders can be treated and which common conditions? What are the causes of subclinical vitamin deficiencies? So of course there's dietary causes, but turns out there's also genetic causes. So for example, if you have a mutation in the transporter, the plasma membrane transporter of a vitamin, for example, you will have intracellular vitamin deficiencies despite having adequate dietary intake. So can we systematically identify these causes and perhaps propose vitamin therapies for these conditions? More fundamentally, what are the novel functions and roles for different vitamins? So some vitamins are well studied, things like niacin is NADH and NAD, riboflavin for FADH, but a lot of the vitamins are actually unknown exactly what role they play in metabolism. So a lot of the lipophilic vitamins, vitamins A, C, D, E, are broadly known as antioxidants, but the exact role in which metabolic pathways depend on these vitamins is still not known. And so we're combining a host of animal physiology with, we're developing novel vitamin reporter tools to measure intracellular vitamin levels. And then we're doing a lot of systems metabolism, genome-wide CRISPR screens to answer these above questions. So the types of things that we're hoping to answer is what are the molecular identities of all the transporters and processing enzymes involved in vitamin metabolism? Are there vitamin sensors like HIF? Are there transcription factors that sense different vitamin levels and initiate these adaptive responses? Can we comprehensively identify all the enzymes that depend on a given vitamin, et cetera? So with that, I'll stop here and take some questions. Thank you to my lab, who's really been chipping away at these sort of big picture questions and more to come in the next few months. I'll stop here.
Well, thank you very much. It was a super neat talk. I mean, I read the papers, but adding more to the story where kind of rounds things out even more. So I'll open up for questions. I have a question while people think of them. So have you thought about looking at primary mitochondrial DNA mutations and their response to hypoxia? Because they're a little different. A lot of them are, in tRNAs and stuff like that.
Yeah, absolutely. We haven't done a whole lot of that yet, and it should be done by someone, if not us. We did, I think, put a few Paul G mice, mutator mice in hypoxia, and there wasn't a black and white result, but I think those animals also just live so long. So I wouldn't say that we answered that question rigorously yet. In the screen, it was primarily complex 1 related mutants that were rescued, not broad ETC deficiency mutants. But more recently, we're seeing that might not be true in vivo, that it might be a bit more general. So
yeah, and I mean, in the same line, like a lot of mitochondrial diseases have a depletion in pDNA.
mm-hmm.
Like there's Twinkle and Pol G, and so I'm curious if those interact in the same way. I mean, there's mice out there to do this. So
yeah, absolutely.
Alessandro.
Yeah. Oh, great to hear about the serendipitous finding when you're just like, okay, let's try and put the mice in hypoxia and see what happens, and then the magic just happened there. Yeah. Almost a self-serving question, but when you looked at the oxygen tension and the whole body oxygen consumption and the oxygen tension in the brain, did you look also in other organ systems? Is this something like just pervasive, or is it specific to the brain?
Yeah, we've just done a little bit of that recently, and at least in the lungs, it seems to also be the case. And sort of more generally, theoretically, it should be everywhere if the whole body oxygen consumption is decreased. In mitochondrial disease patients, there's, I think, 1 or 2 studies looking at venous oxygen tensions. And those are higher in mitochondrial disease patients, again, suggesting that it's a whole body phenomena. Some organs might be able to compensate better than others, right? So for example, if you have brain hyperoxia, there's some evidence that blood vessels can actually constrict to counter this effect, but clearly that's not enough in our animals. So basically we think, at least in the brains and probably lungs, that's the case, probably everywhere. Yeah.
Thank you.
There's a question in the chat by Paul Lianza. Says, regarding therapies, thinking on the slide showing clinical trials, do you think oxygen perturbations would need to be chronic/lifelong regimen for patients with mitochondrial diseases, or would treatments be curative?
Yeah, good question. I should have talked about this. Yeah, we think for the most part, at least in the animal models, it needs to be chronic. And so we've done the experiment where sort of after 6 months of hypoxia treatment, the animals are looking great. We put them in normoxia and they crash within days. And so that's unfortunate. It'd be nice if we could just sleep in hypoxia, live in normoxia, or have it be about treatment. But yeah, unfortunately, it seems to be needed chronically.
It looks like Peter Rabinovich has a question.
Okay, Isha, that's a great talk. You're finding that there is hyperoxia in these complex 1 knockouts is really interesting. And I think for most of us, the initial reaction would be, oh, well, okay, this means that it's reactive oxygen species that are guilty, and let's think about antioxidants as a therapy, which actually doesn't work as well as what you've shown. And you suggested that reactive oxygen species weren't the main culprit. So I wonder if you would speculate as to what you think is going on.
Yeah, we're still working on this, and more to come in the next few months, hopefully. But oxidative stress might be a part of it, but we don't think it's sufficient. We think that molecular oxygen itself might be oxidizing certain structures, and so it might be the case that superoxide, increases the degradation of certain proteins or lipid peroxides, et cetera. But we think that molecular oxygen has different sort of oxidative tendencies compared to superoxide or hydrogen peroxide that cannot be rescued by antioxidants. So we're finding that certain protein structures are basically destabilized in high oxygen. That's our current hypothesis.
Great, thanks. Looking forward to hearing more of that.
Yeah.
Any other questions? Well, if not, I'd like to thank again for giving us a really neat talk. I'm looking forward to hearing how this continues, because it's certainly really cool stuff.
Thank you. Yeah, let me know if anyone wants to work on hypoxia and mitochondrial DNA mutations or anything else. Happy to chat.
All right.
All right.
Bye, everybody.